So thank you to join us to this special edition of Web Biomat focused on women in science. Before we start, I will remind you few information. As most of you know, we have a conference in the Croisic for Biomat conference, and the deadline has been extended. So don't hesitate to register because the places are limited and we soon reaching that limit. I will also remind you that our final web biomat on women and science take place March 25 with Stéphanie Descroix in Sorbonne University. And now I'm pleased to introduce Dr. Claire Dessalles, who is assistant professor at Light and Matter Laboratory at the University of Claude Bernard Lyon. She presents a presentation entitled From Cellular Mechanics to Tissue Physics, a career at the interface. Claire, the floor is yours.
Thank you. So first of all, good afternoon, everyone, and thank you for the invitation. It's really nice to be able to present to this Biomat group, which I think is a really great initiative. So let me start by sharing. So feel free to interrupt me at any point. I think there will be a dedicated question time at the end, but if there's anything that is not clear, if you'd like a precision or you want to discuss something ongoing, I'm also very open for this. And so during my talk, I will do a mix of science presentation in a more seminar format, but also sharing some more personal information about my career, how I made the different choices, why I did them, and a little bit of the behind the scenes for the different projects. I will also briefly touch upon, because I think it was the theme for the month of March, the more women in science aspect. So to start, I will give you a brief overview of my career so far. So I trained initially as a mechanical engineer at École Polytechnique, after which I did a second master's in bioengineering at PFL. And because it was a double diploma, I had to do 2 internships to validate both masters. So the first one was in Germany on the mechanics of biological tissue, and especially more specifically cardiomyocyte tissue, so heart cells. And the second one was on the mechanics of 3D tissues also, but in this case, they were synthetic. After that, I did my PhD for 3 years at École Polytechnique in France, working on the microstructure and the mechanics of monolayers. I then went on to Geneva to do a postdoc on tissue nematics. And I started a permanent position at CNRS, so hosted at the University of Lyon 1 year ago, 1 year and 1 month ago, exactly. So you can see that throughout the different projects that I worked on during my career, all of them have one thing in common, or multiple things in common, actually. They are very often 3D structures, so complex in vitro systems, mixing physics, mechanics, and biology, and in particular systems that can be actuated and that dynamically deform. And so this is really what I'm interested in, is trying to understand the dynamics of these complex systems thanks to in vitro models. So the first one, so this was really the first discovery for me of research in academia, and it was one of the main reasons why then I stayed for a PhD, because I fell in love with academic research mostly because of this internship. So the idea was that we suspended cardiomyocyte cells in a gel, placed it inside these little boxes that had 2 pillars inside. And because of the contractility of the cell, they will compact the gel and create this tissue that will be wrapped around the 2 pillars. We use the schematic side view here. And because these cells are spontaneously contractile, if you look at this under the microscope, and this is a real-time recording, you see the tissue beating, really like a heart is doing currently in your body. And so the task that I was working on was to first develop an external electric pacemaker. So to make this tissue beat on a specific frequency and on demand, and to then characterize the forces that are exerted, because if you know the properties of the elastic pillar, you can extract the force from the deformation and compute this as a function of the microenvironment of the tissue. So the second internship that I did had a very similar idea of something that was deforming on demand, except this time it was a synthetic hydrogel that had nanorods embedded in it, as you can see in this schematic here. And when you shine a laser on it, the gold heats up, warms the polymer, and the polymer becomes hydrophobic. So basically, it's when you shine the laser on it, it squishes all the water out of it and it shrinks. And it's what you see in this movie. So these are recordings that I performed at the time of trying to understand the dynamics of the system and to relate this to pore elasticity of the dynamics of the fluid getting out of this. And the second part of that project was to then try to build micro machines, because if you have this, you have really an actuator that you can control with light. And so by using this as a local motor, let's say, and combining it with a backbone, we could try to do like microscopic tweezers. So this was really the most simple design, but then they have explored more complex one to be able to close, open, rotate, and then interact with living tissues using these micro robots. And so based on these two internships, I realized that really I liked the process of academic research, the curiosity-driven approach, the doing the experiments. So this was really important for me, and discovering and trying to understand. So really like a detective's job, trying to unravel the mystery of a murder, or like trying to find the clues that will help you understand what is happening in the system. And I also realized that I had a preference for non-synthetic materials, so living ones, because they have this natural activity. They have this spontaneous behavior that we're trying to understand. That was fascinating for me. And they have also the benefits that they are understanding their behavior is tightly linked to the medical field and the potential ability to help improve people's life. So this was a bit what I had in mind when I decided to continue and to pursue a PhD and how I chose my PhD. And yeah. And so for that, I came back to one of my professors from my time at École Polytechnique, Professor Abdul Barakat, and he was working on the mechanobiology of the vasculature. So the human vasculature is an extremely complex system. What you see on the left is an actual real human cast, and you can see that you have a very intricate network of vessels of very different sizes that course through your whole body and that are extremely dense in some specific organs. Like, for example, you can see clearly here the brain, the lung, the kidneys, etc. They're present throughout and they're essential for your survival. If you zoom into the structure of these vessels, you go from very large ones that are centimeter scale with very thick walls that are millimeter scale down to the microvasculature. And this was the system that I was interested in for my PhD, and in particular because they're composed of just one layer of cells that sits on top of a basement membrane. So you really have a simple system that performs essential functions, and we wanted to try to understand a little bit more about it. And for that, you can look at many different aspects, of course, of endothelial cell function. But because I was interested in mechanics and physics, and this was also my background, we wanted to understand how the different forces of the vasculature play a role in the physiology of this monolayer. So this is a brief overview of the different forces you can find. So you have all the contact direct stresses, so the forces that the cell sense from the underlying matrix, and all the flow direct stresses. And I was really focusing on the flow direct one. In particular, the deformation, which has been much less studied than, for example, the shear stress, which has attracted a lot of attention. And for that, as I mentioned before, I work with in vitro system. They're great because they're highly controllable. You can really tune the microenvironment of your system to understand the role of a specific input on the tissue of interest. And so I developed a microvessel on chip. So organ on chip are a class of in vitro systems that are becoming very popular because of their versatility, cost, and easy fabrication. And so they combine different aspects that are presented here. Like very often they will have a microfluidic component, special attentions to the material. They will allow 3D culture of multicellular cell types. They can even have sensors for real-time monitoring and the ability to do mechanical stimulation, which was what I was focusing on. So this is an overview of the system that I developed at the beginning of my PhD, which is a microvessel on chip. So it's really a tube inside a hydrogel. So what looks like water here, it's actually a hydrogel, collagen hydrogel. And you have a tube inside of it that is lined with cells, and you can perfuse this tube. Because of the flow, you will have a friction force, which is called the shear stress acting on the cells. And you will also have deformation because if you increase the pressure inside the tube, it's really like a balloon, it will deform and blow up. So what was unique in this system was the ability to deform the monolayer of cells. And this was thanks to the hydrogel that I used, because a lot of organ-on-chip systems are based on harder, stiffer materials. And this was part of, let's say, a new branch of organ-on-chips, which are soft or hydrogel-based organ-on-chips. And this is thanks to the pore-elastic property of, for example, collagen hydrogel, which dictates the dynamics in my system. So this is an overview of the whole system, what it looks like under the microscope. So what you see here in the center is the tube of cells that is embedded inside this hydrogel. And here you have the outline of the PDMS box that's around that holds the gel. And that will enable you to do all the microfluidic connection. So to fabricate this, so basically when I started my PhD, the project was completely new. And so my first task was to build a system that will allow us to impose these forces on the cell. So the first question I had to decide was what kind of fabrication technique I was going to use to make this system. And I decided to go with a templating technique because it was cheap, didn't require any complex systems like 3D printer or laser carving or more advanced ones. Because in my idea was if I manage to make a system that works, that is interesting, it would be nice if it's accessible to the community and in particular to the biologists who don't necessarily have advanced microfabrication techniques. So this is why I chose this templating approach. And so the idea is very simple. You have a PEMS housing, you have a needle, you pour the gel around the needle, and once the gel is polymerized, you pull the needle out and you have a hollow tube inside your gel, which you can then seed with cells. And if you let the cells grow and then you stain and image them, you can get this kind of images that reveals that you have a confluent monolayer with adherence junction connecting the cells and a cytoskeleton that has an organization that is close to what we expect. So once I had developed the system, the first thing we did was to characterize the dynamics of what we could do inside of it. So basically by controlling with syringe pumps the flow inside, we could do complex flow patterns and complex deformations. So for example, here you have a real-time movie on the top left that shows you beads flowing in the channel. I put some beads to be able to track the flow. And this is the flow rate as a function of time that is presented on the other panels. And this also helps you appreciate that the deformation is dynamic and reversible. You can really have a vessel that beats and that pulsates like the ones in your body. So I spent quite some time characterizing the dynamics of the system. So what kind of shear, stretch, and pulsatility we were able to reach. And the important thing was to match the physiological values because what we want to do is to impose to the cells the same force as what they feel in the body. And I then explored potential ways to tune this. So the impact, for example, of the gel concentration, the dimensions of the housing, the flow rate, or even if we add an additional resistance at the outlet. And based on this, we did the first paper that was really focused on, so a bit more method-oriented, understanding the dynamics of this hydrogel-based and hydraulically actuated system. So from then, I had a working system and I could try to look at what the cells were doing. And I decided to measure or to try to measure monolayer elasticity. Because if you think about it, I'm applying a force through the pressure that is deforming the system. And if I measure the deformation, I can record what are called stress strain curves or force deformation, which can give information about the mechanical property of the system. So for that, I did a swelling assay. So basically you increase the pressure rapidly. So in 1 minute, for example, in my case, and you record the deformation. And the recording can look something like this. So you see that it will look back, but basically at the beginning, you have a rapid inflation of the tissue, and then it slows down and continues to deform slowly, even if the pressure is still linearly increasing. So if we plot the deformation as a function of the force, we can have these stress strain curves that I mentioned, from which we can extract, for example, the Young modulus, which is here the slope at the origin. And we can see that we have more complex nonlinear behavior. For example, in the case of the yellow curve, which is on the less concentrated gels, we have what's called strain stiffening, which is the fact that the monolayer becomes harder and harder to deform. One of the things we did was to vary the gel concentration, and we saw that it impacted, for example, the structure of the actin network, and that this in turn fed into the young modulus, that the cells on the more concentrated gels were stiffer than the cells on the softer gel. And this is a typical example of mechanoadaptation. So this is really instantaneous mechanics. So when you do a deformation, you just measure the properties. And then I decided to look at more long-term mechanoadaptation. So if you're a cell, an endothelial cell in the body, and then suddenly the person has hypertension, there will be a pressure increase in all the blood vessels. Will the cells respond to this? This can also mimic, for example, the onset of the heartbeat, which will pressurize vessels that were not pressurized before. So basically any variation in pressure in this otherwise homeostatic system. So to do this, instead of doing a slow ramp, I just increased the pressure instantaneously and then maintained this over 2.5 days. So to do this, I use a very simple technique, which is called the hydrostatic pressure control. Because if you control the height of the pressure at the outlet of your system, this determines the pressure here. And the difference in height between the inlet and outlet will set the flow rate, because if you have a flow rate equal to 0, these cells die. So you need to have a minimal flow rate with a very small shear to keep the cells alive. What this looks like in real life is my little microfluidic system with straws. So these are actual plastic straws because, again, they're very cheap. And they allowed us to have a system that was easily reproducible and easy to control. And as you can see, you have the outlet height that determines the pressure. So for example, here, 8,642, and the little difference between the two, which will be the flow rate, feed the cells inside. And you can put this setup in the incubator and then have a look at how the cells dynamically respond. And so you start by a monolayer of cells that is normally longitudinally aligned because of the curvature. They want to avoid the curvature of the channel. And the question was, what happens if I pull on them in this circumferential direction? So if you apply stretch, after 7 hours, what you see is that the cells have changed orientation. They have now elongated in the direction of the force, and the nuclei have followed. If you look at the cytoskeleton, you see the same pattern. So these are actin stainings, and you can color code it by orientation, and you can see that the cells have completely reoriented. So this was where I was at the end of my PhD, with basically having built a system, characterized what it does, and done a first preliminary set of experiments to understand or to explore how the monolayer responds to force. And now I want to give you some more behind the scenes of how this whole process actually came to be, and maybe a couple of tips for those who are still doing a PhD. So the original title of the PhD topic that I got in was the role of mechanics in Alzheimer's disease. And so why in Alzheimer's disease is because I mentioned that the brain is a highly vascularized organ. And now there is this vascular origin hypothesis for a lot of neurodegenerative diseases. Because basically each neuron has its own capillary. That's the distribution of how dense the network is in the brain. And now people think that some of the time neurons will dysfunction because it's the vessel next to them that is dysfunctioning, which is perturbing their normal activity. And so potentially diseases like Alzheimer's could be actually of vascular origin, and especially because the vasculature of the brain is very fragile. And it's fragile in particular to pulsatility, because normally vessels in the brain do not deform because the pulsatility of the heart is dampened by the arterial tree. But as you get older, your vessels become stiffer, and so this pulsatility penetrates deeper into the brain and can damage these vessels. So this was what we wanted to explore, so the role of hemodynamic forces on the vessels of the brain. So for that, we needed to have mechanical forces, so with high pressure, and look at the role of hemodynamic forces on what's called the BBB, which is the blood-brain barrier, which is the vessels of the brain that are composed of 3 cell types, so brain endothelial cells, pericytes wrapped on them, and astrocytes, which are cells of the brain, but that have feet that wrap around the vessels. So a 3 culture of 3 different cell types. And because we're interested in the tiniest capillaries, we needed to have a big perfusible channel to be able to impose our forces, but that would be connected to a capillary bed of tiny microscopic vessels that could be generated, for example, through vasculogenesis, which is seeding cells in the gel and having them connect together. So this was the original goal that was given to me. And as the project progressed, I really quickly realized that I would need to adjust what the aim was, which is very typical, I think, in most PhD projects. So the first thing we did was to actually define what forces we were interested in, and that it was a combination of shear, stretch, and the dimensional time aspect with pulsatility. So this was okay, and this was also in the core of what my lab was an expert on. But then when we turned to the biological system of interest, there was no brain endothelial cells, no parasites, and no astrocytes in culture in my lab. So we went to find collaborators and try to understand more these cell types. And I quickly realized that this was way too ambitious and that focusing to start on just endothelial cells and standard easy to culture endothelial cells was going to be enough for this first project. The next step was to realize that having two perfusible channels interconnected with a vascular network, etc., was also very complex, and that maybe we could start with a single perfusible channel, like I showed you. And this is also where we realized that we would need to have a look at the pore-elasticity of the hydrogel, so slightly different field to understand the dynamics of our microsystem. And so this is really how I ended up with the PhD that I just presented to you from the initial one. And this was a trial and error. This was talking to collaborators, and this was doing a lot of literature to try to understand how difficult it is and the added value of doing it ourselves compared to what was already there. And in terms of timeline, what this looked like was basically I spent the first year building the microvessel on chip system. I had my first monolayers that grew with living cells, I think in April or May, which as a PhD student can be very scary when you are 8 months into your PhD and you still do not have a working system. But then things went really fast because once the system worked, I just spent a lot of time characterizing it, and characterization was not too difficult. It was just running a bunch of different experiments to understand the dynamics. And so I spent most of the second year doing this characterization, preparing a first paper, and trying to understand the role of the gel, which was a big surprise for us. And the third year, I did this investigation of the response of the cells to one of the forces we were interested in, so just a very simple pressure step. And I spent also time doing the revisions, etc., to finish publishing the first paper. So from here, I have a couple of tips. Of course, you know, every scientist has its own bias to their own career. You should, you know, have a collection of advice from different people. But I think these are the ones I wanted to share with you today. And these are the things that I spent a lot of time thinking about during my PhD to try to make the best of it. So the first thing I spent a lot of time on was to try to think what makes us being the team of researcher and the system we could develop, special. Because this field of vessel on chip and endothelial mechanobiology is not new. There are a lot of researchers. There are a lot of big researchers with gigantic labs that have 10 students working on this in parallel that can go much faster than me. So I realized that if I were to do a system too close to theirs, or if I were to ask questions that were similar to what they were interested in, they would outrun me. So I really spent a lot of time doing literature trying to find what combination of expertise and what we could do that would make our approach and the questions we were asking different from what other people were doing. The other thing that I found quite interesting is the difficult balance between persistence and obstination, because you need to be persistent enough to push through obstacles and to try again every time it fails, especially when you have a little bit of engineering and you want to develop a system. But you also don't want to fall on the obstination side, which is to keep doing it when it has no chance of working. And you need to learn when to quit, when to say, okay, this is not possible, or this is going to take too much time, or this is not as interesting as we thought, and this part we stop. You cut your losses and you focus on something else. And yeah, so this balance is quite difficult, to not give up too soon, but to also learn to give up at some point. The other one was to be open to surprises, to what you can see in many papers, which is called a serendipity, all this discovery by chance. And this happens when you stop, you look at your system or you look at your results and you ask yourself, what do I actually have? Not what you wanted to do, but you just look at it, you try to look at it with fresh eyes, and sometimes you realize that what you have in your system is actually more interesting than what you wanted to do, or there's this peculiar observation that the cells are doing this or that that you were not expecting, and that this could be interesting. So to try to have these fresh eyes and be open to what you were not looking for. Learn to read, which is yeah, you. I think you need to do a lot of literature, especially at the beginning of the PhD when you're discovering all this new field, and you can fall in the rabbit hole very easily, where you end up just reading a lot of paper that are useless and spending a lot of time on very little concentration of information. So learning how to read paper efficiently, to know why you're reading this paper, and to focus and to stop when you have reached the outcome you needed from that paper. The other one was to always have a plan, but be ready to change it. I think it's a bit linked to this serendipity, which is for me, I always needed to know what the next 2, 3, 4, 5 experiments would be on the short term, to have a big plan of, okay, this is where I want to go with the initial goal of Alzheimer's, which then switched to something else, etc. So to have a plan that will help you structure your thinking, structure your experiments, structure your timeline, but be ready to change it so you're open to these surprises, which can be more interesting. The last one is more personal, which is in science, one of the specificity of academia that I think we're all familiar with is that you work on very, very long time scales, and you have lots of failures and things that don't work before you succeed. So to really take the time to celebrate the wins and to enjoy them just as hard as you feel the difficult days. And this is, I think, sometimes one of the things we forget to do. Yeah, take the time to celebrate what works. And finally, start thinking a little bit about what makes you special, you being a scientist. So slowly you can start thinking, okay, what are the different techniques? What is the expertise that I'm accumulating that is going to make me into a specific scientist? Okay, one last thing I wanted to mention for my PhD are the other activities and things I was doing. So I was lucky that I got to supervise 4 different master students, and I think this is something that I really advise if you can, and to push for it is to have the opportunity to supervise people, because this is also how you learn to do it, and this is going to be part of your job after, whether you go to industry or to academia. I also I spent some time writing reviews. As I said, you will do a lot of literature. You will have to write a whole introduction for your thesis. Using all this knowledge and transforming it into a review is a very interesting and useful experience. And finally, I had lots of different collaborations. So some of the collaboration, like for example, the first 3 that I mentioned here, are people that I went to bring to my project. So really collaborations to help in my project. The first one was to do laser ablation. The second one, the modeling. The third one to try to understand the porosity of the gel. But I also had collaboration, especially with these last 4, which was because they were interested in some expertise or tools or techniques that I could do. And so this was really good to also broaden my horizons, to see a bit other projects, to get to interact with other people, and to participate in other types of science. And one things to keep in mind if you're doing this is to balance the investment you put with the investments of the collaborators, because of all these projects, only half of them went all the way to a full project and a paper. And so you don't want to spend too much time or to invest too much time on something that doesn't have a future. So you need to be careful to not out-invest the other people in the collaboration. Okay, so from that, the question was, now what? I wanted to do a postdoc because I really liked academia. And the question was which one, because you have many different possibilities. You will have some people that are interested in your expertise because they know you from your PhD, but you can also look for things that are completely different. So in my case, what I wanted to do, also based on advice that I got from mentors, was to broaden my tool belt in the idea of staying in academia after. So to say, I have learned to do mechanics, I have learned to work with vascular cells. now I need to do something else. So I want to change the cell type, and I want to switch from mechanics and rheology to something else. So that something else was physics. And at the time, I realized that in Europe, there were lots of things around active matter, collective matter, and things like this. And I realized that this was probably going to be something that was going to be useful for me to understand the physics of tissues more broadly, something that European labs were very good at, and sort of like the next place to be for me. And I also knew that I wanted to go abroad because this is almost mandatory if you want to get a position in France currently. So this is how I ended up looking for different places in Europe that were doing this collective matter explorations, and I went to do my postdoc in Geneva, in the lab of Aurélien. So the postdoc really, the central question was to try to understand what is the role of physics, and in particular liquid crystals that I will present just after, in shape making. So shape making is the process of morphogenesis, which is really how tissues self-organize into organisms. And although some early events like gastrulation, which involve epithelial-like cells, have been studied quite a bit, there's still a lot that is unknown about later events that involve, for example, muscle cells. So how all these organs that I underlined here, they are all surrounded by a layer of muscle cells, and how do these organs take shape? And if you focus on muscle cells morphogenesis, one of the things you see is that it's highly collective. You have cells that are elongated, and you have very high forces. And if you show these three ingredients to a physicist, what they will think about is liquid crystals. Liquid crystal is a state of matter that's between a solid, because so you have the crystalline aspect that is shared, and this is because your particles have orientational order. They will tend to orient like each other. And it's like a liquid because there's fluidity, the particles can move around. So a key feature of liquid crystal is orientational order, the fact that the particles orient together. And the consequence of this is that this leads to anisotropic forces. The forces that the cells will exert will be directional in the direction of the cell orientation. So I started my postdoc by doing 2-3 months of literature. And the more I was reading about liquid crystals and living liquid crystals and things in cells, etc., and then suddenly it made me realize that elongated cells and application of forces was directly linked to the end of my PhD and this set of data that I did and never really made anything with. And so basically at the beginning of my postdoc, I went to my supervisor. I asked him if he was okay with me spending a bit of time working on PhD related things. He was very supportive. And I spent a lot of time reanalyzing all my data from the PhD that I showed you. So for example, the fact that the cells reorient using this framework of liquid crystals. So this Q is the order parameter to quantify the reorientation of the cells and to understand whether this would allow us to understand the mechanics, because now I knew that the orientation of the cells was related to the direction of the forces that they exert, and that maybe this was what the cells were doing to resist the force that I was applying. For my postdoc, I also had a collaboration with theoreticians, and the University of Geneva has many different theoretician groups that are excellent, and this was a very good environment to explore this. And so I actually went and contacted Guillaume Salbreux, which was at the time a PI, is still a PI in Geneva, to try to see whether we could do a collaborations to better understand what it was doing. I quite like this citation. It's a bit provocative, but that all models are wrong, but they're useful because they help you rationalize your data. They help you understand whether the hypothesis that you have has some quantitative truth. And we ended up building an active matter model that was balancing the force exerted by the pressure on the cellular tube by the force exerted by the tissue that was depending on the alignment of the cells. And so the points here are my experimental data. The lines are the things from the model, and we were able to connect the two. And thanks to this project, at the end of my PhD, really evolved into something bigger that we ended up publishing 2 or 3 years after the end of my PhD. It really turned into something quite different. But I still had a postdoc to do, so back to the postdoc. And here I want to highlight this, which I think was for me the biggest difference between the PhD and the postdoc, was that during my PhD, I only had my PhD. Some side project collaboration, but really I had one focus. Whereas during the postdoc, you start to multitask You start to have things left over from your PhD, collaborations left over, your main project. You're also supposed to be involved in the projects of other people in the lab, which meant that you need to divide your time efficiently, and it can really feel like you're just failing and screwing up all the different projects in parallel. So yeah. Yeah. So I think for the interest of time, I might go a little faster over the postdoc, so we have time for discussion also after. But yeah, so one of the things that's important in liquid crystal is the presence of topological defects. So because these particles are not always perfectly aligned, you have points, like here in blue and red, where you cannot define the local orientation, which are called topological defects. And they are related to the nature of your liquid crystal, whether it's nematic, so you have no front-back difference, or polar, if your particle are different from the front and the back, and that means they will do different topological defects. And why they're called topological defect is because they're linked to the topology of your system. And why this link? It's because if you have a surface that has a topology similar to a sphere, the sphere has a charge of 2. So that means that you necessarily have topological defects, and the sum of these defects has to be 2. Otherwise called the hairy ball theorem. If you put hair on the ball, you cannot brush them perfectly. You will necessarily have singularities like this, for example, which is 2 plus 1, and 1 plus 1 is 2. Okay. Okay, so why they're interested? Basically, in the past decade or 2 decades, people have realized that these defects are important for the development of tissues, and they have shown this in vivo in the Hydra organism, or in vitro, for example, with the assembly of muscle cells. So what I was interested in was to try to understand the role of topology and curvature in this process, because all the studies so far that have looked at living tissue through this lens have focused on flat substrate. So what I did is I put cells on the surface of a bead, I let them grow, and I followed this over multiple days or even weeks, praying for no contamination. And what we see is that there's a first symmetry breaking event around 1 week, where the cells start to accumulate at the 2 poles, and that these poles are stable for a certain time until the second symmetry breaking happens and they start to fuse together. So this is a very complex process that involves many steps. We decided to focus on the first symmetry breaking. And this was interesting because although this is not what muscles do in your body, it recapitulates features of morphogenesis. So symmetry breaking, spatial temporal patterning, multilayering of cells, collective behavior, etc. And we wanted to see what was happening in the nematic organization of the cells, and in particular the actin. So a big part of my project involved image processing, because if you want to image these structures, you will have a complex multi-layer structure in the 3D environment. So we spent a lot of time developing, for example, this tool that allow you to measure the thickness or the curvature of your tissue, which is called NEMO, and this tool will be published soon. So if you're interested in this, we can, feel free to contact us. But most importantly, what NEMO allowed us to do was to extract the orientation of the cells on this surface. So we were able to look layer by layer at the alignment of the actin network, as you can see with the vectors here. The color corresponds to the order, so whether the particles are aligned like the neighbor, and to find the topological defects from this. And then we could study the dynamics of the defect. So initially you start with 4 halves, and yeah, I'll probably skip a bit this. And then as the tissue multi-layers, you see a complex relative orientation between the layers. So for example, this perpendicular orientation, which is not predicted by theory and that we're exploring right now. And then you see that at the same time as the tissue is forming these mounts that I showed you, you have fusion and binding, or binding and fusion of the defects. So you have this central region that is very ordered, and the topological defects are now colocalizing with the thick part of the tissue. And so we're trying to understand how the nematic field evolution drives the collective behavior and migration of the cells to lead to this accumulation of cells at the poles. So if I summarize briefly this project, basically we have decomposed the evolution into 3 main features, and they all match events that happen at the nematic level. One of the things we're currently working on is the dynamics, so trying to extract cellular flows from this multi-layered structure. And this is still ongoing work. Okay, so for the postdoc, I try to also do a little list of tips. Of course, all the tips from the PhD are still true, but now I think you have things that are more specific to the postdoc step. So one of the things that I found really important was, and difficult, was to prioritize. Because as I mentioned, you now have a lot of things to do in parallel. You cannot do everything, and you need to be strategic. You need to prioritize between what's really important and what is just like, okay, and can wait or things like this. And to be able to prioritize strategically, you need to decide on your future step, what you want to do after. I find that a lot of postdocs want to wait until the postdoc is finished to know how successful or how big their paper was, etc., to decide. I think it's really good to really think about it early and to be strategic to get there, because you will not orient your postdoc the same way if you want to go in academia, if you want to go to industry, if you want to start your own startup. The things you will focus on, the collaborations you will start, the classes you can take, etc., will be different. So to prioritize based on what you need to achieve this next step. And to be able to know what that is, I find it's really important to talk to people and to mentors that you have potentially developed in your PhD or the postdoc, or people at conferences, etc., really early. Do not wait until the end of it. Start the first year of your postdoc and try to understand what is the path for the next step, like what you need to do, what you need to achieve to be able to unlock that next step. So with that, you will start to build a network that is both mental on more career development, but also a network on science. And this network is going to be useful for the day if you want to transition to a PI position, because this is also going to be your first round of collaborators. Continue to mentor people. Will be easier now that you have more experience. Would be interesting if you can be involved in, for example, the project of a PhD student in your group, to continue practicing these skills and to develop this ability, because it's something that you become good at. It's not something that you just like good or bad at initially. And I think to summarize the difference I find between the PhD and the postdoc is the PhD, you're really learning to be a researcher. You're learning to do research, to develop a project, to try to understand the steps to follow it, etc., to analyze data. Whereas the postdoc, you're learning to be a PI. Normally, you kind of know how to run your project, and now you're learning all the rest, like how to multitask, how to start thinking about applying for grants, like a fellowship for your postdoc, how to start interacting with other people, going to conferences, presenting, etc., etc. And the key question you need to be able to answer at the end of your postdoc, especially if you want to pursue a PI position, is who are you as a scientist? What's your identity? What is the research you want to do? What is the intersection of expertise that you have developed? And this will help you build your scientific projects that you want to pursue and that will be necessary for application. And so the last transition that for me was a year ago, even though the whole process took like a year between applying to positions, etc., is to switch to now being an independent researcher. And so the first thing that came to my mind was this taking a leap, jumping off a cliff, because really it's the first time that you're now on your own. You can still have, and you should have, a network of mentors, people that can take time for you, that can help you, and collaborators, etc. But really, you don't have a supervisor anymore. There's nobody to tell you whether you're pursuing the right idea or not, whether what you're trying is correct, to give you pointers, to also make sure that you continue to work. You really have to drive yourself and to take all the decisions by yourself, which is extraordinary and exhilarating and very exciting, but also very scary. So for me, the best analogy was really this jump. And it's really next level multitasking, because now your job, you really have different hats. You have the hats that is a pure researcher, still doing some experiments, the one that is applying for grants, the one that's writing papers, the one that's mentoring, the one that's trying to hire students, the one that's going to conferences, the one that is organizing conferences, etc. And so I find that this is really a privilege and really, really fun to be able to have all these new tasks. But your job changes and you need to learn all of this. One of the things I do want to mention, especially because of this theme of women in science, is that I did have a baby during my postdoc. and I'm actually currently pregnant with my second kid. So I did not wait until I had a permanent position to start, let's say, advancing on my personal life. It did come with some challenges. I'm very happy to discuss this further in question if some of you are interested. And so to wrap up, I just wanted to give you a little overview of the research that I want to do now in my own team, which is still going to be at the interface of mechanics, physics, and biology. And it has this double aspect that you might have noticed was present during the different projects I did. On one side, engineering new systems, like for example, the microstretcher, which allow you to ask new questions to try to understand the fundamental behavior of tissues. And thanks to what you understand, you can then improve the systems and gain new complexity in your microsystem. So I really like this double aspect and the dialogue that there's between them. And the bonus is that it allows me to interact with many different fields. So on the engineering system side, you have this direct link to more biological and medical and applications, which gave me the opportunity to launch multiple collaborations, in particular around surrounding the microstructure. And then on the more fundamental things, you have, for example, collaboration with theoreticians, physicists, etc. And I think it's really a source of inspiration, of ideas and innovation to be able to interact with these different people. So with that, I want to acknowledge all the people. So what supervisors, interns, and colleagues and funding for my PhD. Same thing for my postdoc and current collaborators now at CNRS. And I do want to mention that because I'm starting my team, I will be hiring. And so if any of you is interested in PhD, postdoc, or anything, I'm happy to discuss. Thank you.
Thank you, Claire, for your very interesting presentation and all of the tips. We have some question on the chat. Thank you a lot for science advice and the amazing talk in general. I have a question regarding the range of frequencies that you manage to reach by making vessel bits. Does it reflect physiological value, and also have you witnessed any signs of fatigue on cell rearranging during the building process?
Very interesting question. Multiple sub-questions, actually. I explored the range of frequency. So lower is easy. You can go lower, as low as you want, because then it becomes like a very slow pulsation. When you go up to 1 hertz, which is close to a healthy heart, let's say, 60 beats per minute, you get nice amplitudes. But if you go higher, what I saw is that I think there is some fluid dampening in the microfluidic network, and basically the amplitudes start to get smaller. I went all the way up to 4 or 5 hertz, but then you get like really small oscillation and you cannot do the full range. One of the things I haven't tried is that I could compensate for this by aspirating. I only did positive pressure oscillation like you have in the heart. But if I were to aspirate in between, I think I could probably force the system to. So if I impose, let's say, 10 times the amplitude that I would like through the dampening, I might reach the amplitude that I would like. So this is something we can explore in the future. So yes, it matches physiological value, and it can match physiological values for heartbeat, but also for the breathing. Because one of the things I would like to do in the future is to go away from pure vasculature and to just think about it as a microstretcher. And we could also stretch other monolayer like epithelial cells of the lung, and then you can make something breathe like this. And so for the fatigue, so you mentioned cell rearranging, etc. So this takes some time, and I haven't done so far oscillatory stimulation. We only performed oscillation to show that it was possible on like short time scale, but I never monitored the response of the tissue for this. I'm actually writing a grant exactly on the topic, and that is of course part of the future.
Thank you. Another question is building on the previous question. I am interested to know how fast cells change their orientation once flow is introduced, and how this will look like with a rhythmic flow. Would they oscillate orientation?
Yep. So how fast? So we looked at the dynamics in detail in the paper. If you want to check, the speed of the response is also dependent on the amplitude. So if you apply a higher force, they turn faster. There's probably a higher limit to this. They cannot turn instantaneously, but basically they turn in hours. So the between this and this, we had 7 hours for the highest pressures. And after a couple hours, you can already see they are like, in transition. And how would this look with Rhythmic Flux? This is one of the things we would like to do. If I had to, also depends what Rhythmic Flux you do, because if you do purely positive, that means that you have a step on top of which you have oscillation. So you will probably have a combination of the response to the pressure step like I did plus oscillations. That's why one of the things I want to do is to do oscillations that are centered around zero. So pushing and aspirating alternatively, because also aspiration, so how would the cells respond under compression? We don't know. So it's really hard to guess, because my system, yeah, is quite different from what exists in the literature. So really, really hard to guess and really curious about it.
Thank you. Another one was, based on your young research experience, would you like to develop project with medical outcome that require perseverance over 10 years?
Um, so I guess as a PhD student, I probably would not have liked it because my PhD being 3 years, you also are happy if there's some outcome. That being said, you can be part of a bigger project. So there are now funding schemes that are quite big over 5, 6 years. And then your PhD can be one of the work packages that's like one step of this. So what you're doing has a nice question that can be answered in 3 years, but is still a building block for something bigger. Now, for now, yes, I'm actually part of, so we just got a funding to develop a endometrial cancer model on a chip. So the funding is for 5 years, and it's probably just the first step that this will need to be continued. I think this is really one of the things that's amazing once you get an independent research position is now you can think on even longer timescales and have more ambitious projects. So yes, I think personally it's something that I would find interesting.
Thank you. Another question is, how do you end up being able to make difference between persistence versus obstination? The barrier seems quite slim to me.
Yep. I don't think there is a right or wrong answer. It's also subjective. So things that can help you, I think, is talking to other people. So having a fresh set of eyes, like discussing with your colleagues, because like sometimes you're too in your thing. Talking to your supervisor, of course. Um, and then it's also a risk balance. You can also, so if the reward for succeeding that specific step is that your whole PhD depends on doing it, then of course you will need to persist much longer than if it was like, oh, this was a fun side idea that I had, I wanted to try. So I think it also depends on how big is the reward compared to the time you spend on it, how important it is to actually succeed with this. One of the hardest thing I had to do was I did one experiment where I put parasites on the fiber. And one of the things people have noticed is that every time you culture parasites in vitro, they lose their correct shape. And normally they should be bumpy. And nobody has managed to make them bumpy in vitro. And one of my idea was that the curvature of the vessel was important. And so I put the parasites on 5 filaments, just like, oh, okay, I'm going to do this as a side project and let's look what happens. The first experiment, I had 5 parasites and the 5 of them were bumpy. And I was like, this is amazing. And I did it again. And then all of them were flat and again and flat and again and flat. And I tried the substrate different. I tried adhesion. I tried everything I could think about that could have been different in that first experiment. And after 2 months, not doing 2 months full time of this, but 2 months of trials, I never managed to reproduce that first experiment. And I decided to give up because I was like, okay, this is cool, but this is not my main project. And I have exhausted all the ideas that I had. So now it's just repeating the same thing, hoping for a different outcome. And I think there's probably a famous citation that says that this is the definition of insanity. And so I gave up. And it still to this day drives me crazy that I do not know what was different in that first experiment. And it's probably something that I will restart one day. But because it was not essential for my main project, yeah, I don't know if that answers the question.
I think. And the final question, regarding the transition from young postdoc to applying for permanent position, how honored felt you at the time when you decided to start applying, and how did you decide on the location or hosting lab?
Yeah. So, okay, I will start with the second part, which is easier. So how did I decide on the location? So in my case, it was driven a lot by personal reasons. So I was married and my husband had a job that he really liked that was in France. So that was the first thing, was that I wanted to come back to France and all my family was here, etc. And then in France, I decided to go to Lyon because I had kids, and both my family and my in-laws are in the Lyon region. So it was like a big help to be able to juggle both research and family, but also nicer for my kid and for my parents to have each other. So this is, yeah. So once I had picked Lyon, I just looked in Lyon the different labs that there were, and there were not so many of them, and three probably. One I found a bit too biological for what I wanted to do because I was really more on the physics side. And then one was doing biophysics, like what I wanted to do, but on more subcellular scale. So they didn't have really the equipments like microscope, etc., that I would need. And when you start a position, you don't have a lot of money, especially at CNRS at the beginning. So you need to be able to have the tools you need to do experiments there already. And so this is how I decided on the other one. The final check was the people. So I spent a lot of time talking to them, but to other people to have opinion about them. And the conclusion was that it was a really nice, humane, and collegial group, which was what I was looking for. I did not want to go into a super highly competitive place where people are just like trying to outcompete each other. I wanted a place that would be supportive. Yeah. And so for the first part, how ready I felt. So honestly, I started thinking about applying already at the end of my PhD. Not applying now, but I already had that in mind. I discussed it a lot with one of my co-PhD advisor, and it really helped mature this. So it was a really, really slow process of maturing my idea, my project, what I wanted to do. I did not start 3 months before, like, okay, I want to apply in 6 months or 3 months. So what's my project? The project really, I had at the end of my PhD a lot of ideas of future steps to do. And I was like, okay, I could do like research based on this. And then I started the postdoc. I opened to this whole new field. This started to merge and to make my initial ideas evolve into, yeah. So it was a very, very slow process. So I don't think the moment I started to apply, I did not feel unready because it had all this time to slowly mature. And I also, if you consider the first time I considered applying to CNRS, I also did not feel unready because I was not applying right now. It was like, okay, this is something I would like to do in the future, and I will start like thinking around it. So yeah, it was, yeah.
Okay, thank you, Claire, for your amazing presentation and the response to the question, and of course for accepting the invitation.
Yeah. So
Thank
thank you for the
you.
invitation, and
Thank
feel free
you.
to contact me even by email or anything if you have follow-up questions or would like to know anything.